Thank you, Eva, for this very nice introduction. In fact, as you will see throughout my talk, I will directly jump onto our speaker we had just recently of Mark, who was talking about notch. But let me start a little bit more smoothly. So as I have worked on tetralogy of Fallot for the last 20 years, I thought that I give you an overview from starting where we started originally and ending up where we have been for tetralogy of Fallot. So when I started to go into TOF, this was the time more than 20 years ago when the Human Genome Project was still going on. And luckily, I was a member, a part of this Human Genome Project. And what you see here on the picture on the left side is a printout. So print out of the human genome. So if we think on the date we have now on digitalization and so on, it's quite really funny for me to look at these. So this is 20 years ago, and I think we have learned a lot, but there are still some things, some general things we should keep in mind when we talk about the genetics. So first of all, this is a diagram or a picture here showing you the heart development in mouse. And why I'm showing this is to remind you that the cardiac development is really complex. And therefore, I think it's very clear that congenital heart disease are the most common birth defects because it is very early in a very vulnerable phase of embryonic development happening. And what you can see very simply here is that you have two different first and the secondary heart field, where originally the heart is developed from. This is here depicted in mouse, and you see that a secondary heart field depicted in green will give rise to the right side of the heart, and the first heart field more or less to the left side. So what makes the heart so vulnerable as well is depicted here, because what you see is the quantification of numbers of cells and volume during cardiac development. And you see below the embryonic days. So we have a tremendous increase in cellular amount, so in cell divisions and proliferation going on at a very short period of time. And these are now depicting all the different cardiac progenitor cell lineages who contribute to the heart. So when we now imagine that, of course, in each of these cells, we have the same genome, more or less, and the development of the different cell types we have in the heart is just made out of differential expression profiles and expression programs, so activations of those genes. It is quite clear that there might a minor mistake in the genome lead to a full bunch of different phenotypes as we see. So here are, I think, particular pediatric cardiologists are very well aware of these. You see different cardiomyocytes. So the atrial and ventricular cardiomyocytes have totally different action potential, and I have different molecular markers. So these are not just genetically on the expression level different cardiomyocytes, which you can really differentiate in a black and white manner. These are also physiologically different cardiomyocytes. So what does it brings us to? I would like to show you this. It's a very, very, very old picture. We published it, generated is 20 years ago. And I placed on the left side as a German heart center, because every patient I ever worked with, I would say 99% came from the German heart center. And we have now the register of congenital heart disease, Register angeborene Herzfehler, which is a brilliant thing to have. But at the time when we were starting to collect the samples, we didn't have that, so we did it at the Max Planck. And I think what is now in our days totally clear is that we need to have a very deep clinical phenotyping connecting with biobanking to identify the subphenotypes. Well, what you see here is just a hierarchical clustering of different cardiac characteristics, which we had used back in that time for clinical phenotyping in a database we had developed. And you see easily that you can discriminate for different kinds of tetralogy of Fallot who have minor differences in pressure levels and so sub-differences. But when we correlate this difference, the tetralogy of Fallot subcases to the clinical genetic features we see, then we might go further to have eventually tools to to have not prevention, but tools to have diagnosis very early to discriminate patients who might go into severe cardiomyopathy cases later on or not. So combining, I think, every different impact, what can happen to the heart eventually leads to the portfolio of congenital heart diseases. So the pink CHDs below should just discriminate or show here different types of cases where you have one genetic hit, like in A, heart disease, and there are very complex cases. So I worked for the last 20 years on tautology of Fallot, and tautology of Fallot, the non-syndromic form we always worked on, can be very simple, I guess, but the majority is really a multigenetic disease, which has also environmental impacts. Starting with the simple one, selection of monogenetic causes of congenital heart diseases. I like to show it, but I don't think, in fact, that this table is correct when we consider the knowledge we have on our days. So, for example, in the second line, you see here cited 2 listed, which was done in my group. But when I look now back to the study we did at that time, we were focusing on particular genes. Everyone did. And when we would redo the study with the same samples, I'm sure that we would have multiple genetic alterations and not only one. So this is decided one story at that time, 15 years ago, and we saw a panel of nucleotide variations, and there were also some associated with tautology of Fallot, but I think redoing this sample analysis, we would find far more genes affected and not only cited 2. So I think in congenital heart diseases, in non-syndromic ones, particularly in TOF, a monogenetic course is very rare. Instead, we have a multigenic architecture. And this is a study we did. It's now also really old, I would say. It's 6-7 years ago. But it has been the first study to show that tautology of Fallot is not a single genetic disease, it's a multigenic disease. And what we did back at that time was not genome sequencing as we all do it now. We developed an in-house made array where we used just less than 900 cardiovascular selected genes and microRNAs to look at. And we had, as you see on the top numbers at the top, we had a very small but very extremely well-defined set of patients. However, with statistical tools, even back then, we could show that there are multiple genes and well-known multiple genes affected in combinations in the different TOF cases. And I think this gave the first genetic architecture. And what we defined at that time was luckily also confirmed in studies who had been far more outwidened and were followed. So just 2 years ago, there was a big whole exome next generation sequencing study where they used 820 TOF cases and the whole exome. It was done by Bernard Keaveney's lab, and they showed, as Mark just also pointed out, that NOTCH1 mutations are the most prominent mutations and genetic affections in non-syndromic tautology of Fallot. And in fact, this NOTCH1 mutation importance we had already shown in 2014. So in our very distinct selected patient group, we have seen 20% of those patients affected with NOTCH1 mutations. And in the far more bigger TOF cohort used by Bernard Keaveney, they found 7%. So I think from different studies, it's now clear that NOTCH1, particularly for non-syndromic TOF, is a very important gene. And I think it will be really interesting to correlate the distinct genetic affection with a distinct clinical phenotype we see. So this is just going a bit broader beside NOTCH1 at this Bernard Keaveney study, as I found also other genes affected. And what was really interesting, they also saw exactly what we had seen before, that there is a multigenic affection of tautology of Fallot. And they found only 1.2% of cases of their big 800-something TOF cohort were affected by the top 9 different genes you see in that table. So the majority of cases were affected by the other genes, and they are affected in a highly combinatory manner. So there are multiple genes who cause in combination tautology of Fallot. Besides having this clear, affection of genes who in the best case are missense mutation and lead to non-normal pathologic proteins, there are also modifier genes who might be affected in a far broader patient population and even partially in the normal healthy population in general, but in combinations with affection of particular TOF genes, then they are triggering the phenotype. And 2 of these genes are depicted here. One is myosin binding protein C3, well known from hypertrophic cardiomyopathy, and there are distinct affections of that particular gene together with other genes who then can be seen in tautology of Fallot. And you see here on the left side in the picture, you see the disarray, the myocardial fibers disarray in one particular TOF case compared to normal control. And this might be interesting for the clinical side because I can well foresee that in the future, screening for these cardiomyopathy genes also in patients with structural defects might be interesting in order to find probably a clinical association with a phenotype or with an early indication to have a more likelihood of cardiac insufficiency in the ongoing phase. And another very interesting gene which we identified as a modifier gene is ACATS. It's a gene of the mitochondrial fatty acid oxidation pathway, and there is a very common mutation which is present in around 20% of the normal human population. But this leads to a reduction in the enzymatic activity of that particular protein. And in combination with others, genetic alteration, this might trigger the TOF phenotype. So now, having done these studies on isolated TOF cases, what we went quite recently last year, we went and looked at monozygotic twins discordant for tautology of Fallot. Discordant for tautology of Fallot, this you see here in the left-hand side. And what we have seen is that we saw, of course, many genomic variations, but we didn't find genomic variations which were distinct between within the twins and which could explain the TOF phenotype. So this was a very, very interesting and very important, unforeseen, I have to say, observation. And having found that, we went further and we looked at the epigenetic landscape in those twins, because development, as here seen for the famous Waddington development landscape, development can be canalized to follow differentiation by epigenetic modifications and epigenetic modifications like DNA methylations. So what we did a genome-wide DNA methylation analysis within these twins. And indeed, we found very interesting altered DNA methylations. And particularly one which came to our interest was as a TPX20 promoter region, because years ago, in 2018, we had already identified TPX20, a very important transcription factors, to be not genetically altered in TOF, because we did a big screening on that gene at the time, but we found it to be differentially expressed. And what we saw now in the twins is that indeed the promoter region of TPX20 shows a significant differential methylation. So this would go along. So on top of having genetic alteration, I think it's very clear that we have also epigenetic alteration, which might come in as modifiers in triggering the diseases. And this makes a lot of sense if you remind ourselves that I just had this slide showing where we have the different cell types, which we have at the end of the cardiac development in the adult heart, but which have all different origins and the origin cell types, but have common progenitor cells. And from these common progenitor cells, these cells are not driven only by the genetic background, but also by the expression profiles. And there, these differences might be important. So we did also a genome-wide DNA methylation profiling for not only these twins, but also for a broader study of tetralogy of Fallot cases, for far more cases. And out there, we identified SCO2, again, a mitochondrial protein to be differentially methylated in the promoter region. And on the right-hand side, you see it's also differentially expressed. So I think the mitochondria are something to look particularly on as having in mind the modifier ways for defining the genetic architecture. This is my last slide. I always end up with this slide because I think, I mean, of course, we would like to diagnose TOF patients better. Of course, we are always aiming to have a better therapy for TOF patients. But I think what is the final goal? We all want to prevent any type of congenital heart disease. And having said all these epigenetic modifications which come into the development of congenital heart disease, including TOF, we have particularly for TOF the situation that it's well known that vitamin B12 is not only reducing neural tube defects, but it also reduces congenital heart diseases. And the hope would be, and this is in fact true to this very high migratory mitotic index we see in the neurocrests, who are one progenitor cells who travel to the heart, and they have at the very early development of the heart this high level of proliferation. And at that time, of course, you need a high amount of methyl groups where you need B12 for the folic acids, for the folic acid cycles. And the hope would be that besides having this genetic analysis in the future, we might find other ways where we could modify slightly the epigenetic parts of this developmental cardiac developmental process. Okay, so that's the acknowledgement slide. I take the liberty to just have the Heart Center here at that time, because over these 20 years, there have been so many people involved. But the German Heart Center was always the beginning of everything I did. And I just want to point out, maybe majority of you knows, we had again a Springer book where everything about congenital heart disease, clinical features, human genetics, particular and molecular pathways is in. What you see on the left-hand side was the first edition. We are currently working on a second one, which will come out next year. And I'm very happy that Nikolaus Haas agreed to be our clinical editor for the second edition. And if you are interested to read it in a different language, feel free. It is available since half a year in Mandarin. So it was translated to Mandarin and it's published in China. And maybe some of you can figure out on this nice looking pictures for myself what tetralogy of Fallot means. Thank you.
Thank you very much for your presentation, Silke. And I have a question, one question from the audience, and I would really like to encourage the other people in the audience to ask questions. I will ask the first one to you. Have you sequenced patient-derived cardiac tissue, so biopsies from surgery, for example, looking for somatic variants?
Yeah, we have done this several times. We did this really with Sanger sequencing 20 years ago, because the first idea was that somatic variances could be a really important point. And there had been at that time 3 or 4 publications for NKX 2.5, TPX20, and something else. But these could never be, these particular genetic alterations which were seen in these publications, not by us, could never been confirmed by any collaborator we had in a big European project. And we have done also DNA methylation as well as genetic studies I referred to at the end of my presentations, were all done in tissue. And we looked very carefully about it. I think there might be, or there is, everything is happening, right, in biology. So I think there will clearly be very selectively somatic mutations, but these are not the key driver. And these are very, very rarely happening. So we didn't find anything. But yeah.
Thank you, because you already answered my question, which tissue you use for the methylation studies.
Yeah.
But you already said it's tissue. It's not a blood sample. So have you ever compared if blood sample would be possible?
So yes, yeah, and blood sample is possible. So you have to know that blood sample shares around 85% of DNA. This is what we found as well. 85% of DNA methylation profiles. You have similar between the blood samples and between the cardiac tissue. So we used blood samples and we used cardiac tissues, and we compared both. And the cardiac tissue, so we had the same probes used for genetic analysis, for gene expression, and for DNA methylation. So it was all done. It was all done out of the same pool of tissue. But comparing it to the blood, of course, you don't get 100% what you would like to have, but you get really sufficient things. And I think it would be worthwhile to do a far bigger studies with far more patients in the blood for epigenetic profiling, because this is what you.
Yeah, and this is a tissue or sample you can easily get to. And I really think it was important that you said in the beginning, we need deep phenotyping if we really want to differentiate these subgroups also. Thank you very much. I think we have to move on. Oh, there is another question. So maybe we can quickly do this to encourage more audience questions. How these current genetic knowledge could be translated to the patient's routine care, improving their clinical outcomes. Can you maybe briefly comment on that?
I think what is needed is that we do a big study where we do genetic profiling and maybe epigenetic profiling as well in the blood, and very deep clinical phenotyping. So we have to have really deep clinical data. And out of these, I would guess that the outcome will be that there are particular genetic alterations which we can use for the diagnostics, for the decision making, who we potentially have more arrhythmic potential, or who might go earlier into cardiac insufficiency during life. But this is needed before we can have, for non-syndromic cases, I think really something for the clinical daily use.